Greetings to everyone. Welcome back once again to my educational channel on biology. So this video has taken me a long time to produce. Thank God it's done, and I hope that you have time to view it. If not, you just view the experiments that I have given to you, alright? And you can select certain parts to view here, especially like how to write an inference, how to write the hypothesis, or draw the graph, right? And analyze the graph, and then what are the prediction questions and so forth, alright? So this video is also useful for the Form 4 students, okay? Because you've already done quite a lot of practicals and experiments. So Form 4 students also, please view it when you have time, okay? For Form 5 students, do keep calm and study. And if you have any questions, you can ask me within this period of time, this next few hours, before I go and sleep. Alright? So, all the best to you all, Form 5 students. Take care and keep calm and study. So, let's begin. First of all, when we get the paper, we want to start off smoothly. Okay? So, let's see what we need to do. So, take a deep breath, and if it's a guided experiment, any of the procedure, you can heave a sign of relief first. Right? So, let's start off. First, you read the question carefully. Alright, slowly, carefully, and in the midst of that, when you're doing that, look out for the clues. You're like a detective that's looking out for clues and for looking out for hidden treasures. Okay, what are the hidden treasures? All the variables, the objective, and or aim, and so forth. Okay, so the first thing to look for is the objective or aim of the experiment. This is should be at the very top of the question. Okay, at the top of the question, and it starts with the words to study, investigate, or determine something. Right? That is the objective. So underline that, and from there identify and underline the main MV, or circle the main MV and main RV and write on top MVRV. Right? So use a dark pencil to write clearly, and don't use ink. Alright, we don't want to dirty the paper. So everything should be done systematically and neatly. Now next, find or determine what the sub-RV or ORV is. ORV means the operational responding variable. It's the same like the sub-RV. That means the experimental RV. Okay, operational means experimental. So it's experimental RV. What is it? So that is what you need to measure, observe, and weigh as the end results of the experiment. Okay, so the main RV and sub-RV could be different. For example, if you are doing enzyme experiments, maybe the experiment is to determine, the objective is to determine the effect of temperature on the activity of amylase. Effect of temperature on activity of amylase. Okay, firstly, effect of something upon something is the effect of MV on RV. So temperature is your MV, and your RV is the activity of amylase. But how are you going to determine the activity of amylase from each reaction? So the only way to determine it is to find out when the hydrolysis of starch is completed. Okay, and time, get the time taken for hydrolysis of starch to be completed, or time taken for the iodine solution to remain brownish yellow, so that it didn't turn blue-black. That is the time taken for the hydrolysis to be completed. So it's different. Okay, so if this is T, the time is T, the main RV is the activity of amylase is 1 over T, right? So the sub-RV will lead you to the main RV, okay? But the sub-RV is what we measure in the lab, right? Or what we observe in the lab. So you have to still hunt around for the sub-RV, okay? From the procedure. Now next, so highlight or underline these three RVs, these three variables. MV, MV mean RV from the objective, and the sub-RV, which is somewhere inside the procedure. So in the procedure, find out the apparatus and materials needed for each variable, meaning the MV, sub-RV, and FV, and how to handle the MV, RV, and FV. How to handle the MV, sub-RV, and FV. Okay? So underline the parts that have the MV, sub-RV, and FV, and then mark it neatly. Mark with these words, MV, RV, and FV, neatly in pencil. Okay, so apart from that, that means you have found the MV, FV, and RV from in the procedure. Next, you have to find the precautions in the procedure and mark with a P. Alright, so precautions are important to give us more accurate results. Next, if it's a non-guided experiment, take another deep breath and you have to think carefully and come up with the components in steps 5 and 6 by yourself. Okay, you have to think, what is the MV in this case? How am I going to handle it? What is the sub-RV? How am I going to handle it? What is the FV that I must keep constant, right? So do not panic, because normally the experiments will be from the textbook or what your teacher has trained you to do, and you can modify the experiments if needed. All right. So when you get an experiment that you have to design by yourself, then find out how to handle the MV, RV, and FV, and also what are the steps you need to take. Now, what happens if you are designing the experiment yourself in a non-guided experiment? So in your procedure, you have to state how to set up the apparatus, all right, such as labeling of test tubes for enzyme experiments, and then for energy value experiment, you have to pour 20 ml of water into the boiling tube and insert the thermometer into the boiling tube. So all these are steps, right? And for osmosis experiments involving the mass of potato strips, you have to cut the strips into the required length and so forth, or use a cork borer to produce the strips. So the main marks are for how you handle the MV and the apparatus use, how you handle the RV and the apparatus use, and the FV and the apparatus use. Okay, so the MV is what is changed in the experiment from one stage to the other. The RV is what is measured, observed, or the results of the experiment that we get at the end of the experiment. And the FV is what is fixed throughout the experiment. All right, that does not change. Now, next, precautions to ensure safety is very important to write down at least one. So 1 mark for MV, 1 mark for RV, 1 mark for FV, and 1 mark for precautions. And we have to keep to the rules and really carry out the precautions. Okay. For example, in the energy value experiment, you have to stir the water slowly with the thermometer. You must add this word slowly, very important. Because if you just say stir the water with the thermometer, now what happened to one of my students? As I was busy helping another student, this student, one of my students, could be heard stirring the water very vigorously with the thermometer, just like you stir a cup of coffee with a spoon. So you can't treat the thermometer like that. So before I could stop him, the thermometer, it broke, okay, with the sound like that. And that was the end of it. And it's very dangerous if the mercury comes out in the water. Don't touch the mercury at all, okay, because it can be toxic. So stir the water slowly and gently with the thermometer, okay, if you need to stir the water during energy value experiment, okay. And there are other precautions for enzyme experiments and so forth, like you have to rinse the dropper after you test the mixture with the iodine test, right, to test for starch. You have to rinse the dropper every time so as to avoid contamination. All right. Now, for accuracy, you may add the experiment is repeated twice and the average reading calculated if you need to repeat the experiment, but you must show both readings in your results, okay, in your table of results. So firstly, let's discuss the observation, right? So this is the first science process skill that is probably asked in the exam. Now, here are the explanations and the components of each question right now. So when you are asked to write an observation after you have recorded the outcome of your experiment, right, in a table, the observation must have the MV sub RV from the experiment, then the reading or data or value plus the units. So actually there are 4 parts to it. Make sure you don't forget the units too. MV sub RV, reading or data or value, is one item, plus units. So the data is the raw data and no calculations have been done yet on it. For example, if you are getting 2 readings for a particular MV, you must state both readings. Don't go and find the average and then state only the average reading. Okay, because no calculations should have been done yet If there are two readings, you state both readings, right? Plus the units. Then you have to copy the exact MV and sub-RV or operational RV, ORV, from the question if the question is given in a guided experiment. All right. For example, the sub-RV for enzyme experiment on amylase will be, for example, time taken for the iodine solution to remain brownish yellow, or time taken for the hydrolysis of starch to be completed. But some students, if they are lazy, they just write time taken. That's wrong. Okay. So you must write the complete phrase for the sub-RV from the question. All right. And the complete MV that's stated in the question. That's why I need to underline all this in the question. All right. Now, refer to the table of results for MV and RV if the table is given. Sometimes you have to write or construct the table yourself. Then there's no table for you unless you construct it according to the instructions. Okay. Then you will have the sub-RV inside. Now, so let's find out examples of observation. Okay. Now let's look at three observations. All right. The first one is related to the process of osmosis, and the aim is to study the effect of the concentration of sucrose solution on the final mass of the potato strip. All right. So our MV is the concentration of the sucrose solution, which is in this case 0.1 m, and the final mass of the potato strip is the sub-RV, okay, from which we get the percentage change in the mass of the potato strip. And our reading is 2.51 grams, okay, so plus the unit grams in there. All right. Then we also mentioned the initial mass of the potato strip, okay, which is different for different strips of potato, right? So here we have the MV, the sub-RV, and the reading plus the units, okay? So the main one is the first statement. Second statement is just additional. Now, for the another experiment, determining the energy value of different food samples, all right, this is our observation. For groundnut, the final highest temperature of the distilled water when the groundnut is burnt completely is 73 degrees Celsius. And here we can write in brackets, the initial temperature of the distilled water was 30 degrees Celsius. Okay, so there's the rise in temperature to 73 degrees Celsius after the groundnut is burnt. So groundnut is the food sample, it is the MV. And the final highest temperature is the sub-RV, from which we will calculate the energy value. Okay, and then so the value or the reading is 73. The units are degrees Celsius. Okay, so again you have the components MV, sub-RV, and reading or value. Okay, with the units. So this answer is complete. Now, another type of answer involves a color change. For example, for food tests. All right, there's no measurement involved, no units like values, no values involved. Okay, so in test tube A, where lipase is present, the phenolphthalein indicator changes color from pink to colorless after 5 minutes. Now this experiment is to study the digestion of lipids by the enzyme lipase. All right, so in test tube A, the MV is the presence of lipase. So where lipase is present, now the RV is the change in the color of the phenolphthalein indicator. So phenolphthalein indicator changes color. Okay, so color of phenolphthalein indicator is the RV, and the observation is that it changes color from pink to colorless. Okay, so this is color change that is involved in food tests. Next, we come to the inference, which is difficult for some students, right? So pay attention here, and I'm going to explain to you very easily how you can give a good inference. All right. So I'm going to give you the format of the inference. Firstly, we must understand that the inference is actually an explanation, the explanation for the observation that you've stated above. And that means it's the reason why an observation is like that, right? So you have to compare the different measurements or observations for the different MV, right? So we must mention that the sub-RV is higher or lower, shorter or longer, make the comparison. There are no marks for this, but it will help you to explain. Okay, then you put because. Now, we know at that particular MV, the sub-RV is like that. So state the effect of the MV, okay, by giving a biological explanation, the effect of the MV on the RV. And that's how the RV affects the sub-RV. That means you are linking the sub-RV to the MV and the main RV. Okay, so let's look at some examples to understand this format. All right, compare the sub-RV with the other sub-RVs and then state whether it's higher, lower, or shorter, longer, and then because, state the effect of the MV by giving a biological explanation. And so this causes the main RV to be high or long or short and so forth, right? And that affects the sub-RV. That's why the RV sub-RV is like that. Okay, so let's look at an example. Now let's look at another one. So the second observation was for groundnut. The final highest temperature of the distilled water when the groundnut is burnt completely is 73 degrees Celsius, right? Now how are we going to explain that the final highest temperature is 73 degrees Celsius, which is considered high compared to the other food samples, So again, your sub-RV, the final highest temperature of the distilled water is high, right? Now let me put in the components first. Okay, RV here. So what is the reason for the fact that the final temperature of the distilled water is high compared to the other food samples? So it's something to do with the MV, to do with the groundnut. What's the content of the groundnut, right? That affects the energy value, which is your main RV. So the main RV is always related at the end. Okay, it always comes in at the end, at the end of your inference. Right, try and write the answer, and then I'm going to discuss it. Pause the video for a while. Let's look at this. The final temperature of the water after the groundnut is burnt is higher than the others, than for other food samples. Because, now here's your main MV, the groundnut contains a higher lipid or fat content. 1 mark. Releases more energy, fats releases more energy when it is burnt, right? And then this is because the groundnut has a higher energy value, okay? So which comes first? First, the groundnut has higher lipid or fat content and release more energy when burnt, okay? So that makes it have a higher energy value, okay? That's why the final temperature of the water after the groundnut is completely burnt is higher. So the biological explanation is the second and third point here, and also the fourth point, right? So you must relate it to the fat, higher fat content or higher lipid content in groundnut. Okay, as we know, groundnut contains a lot of oil, and sometimes the oil is extracted, okay, to form to get the groundnut oil or peanut oil. So just now we discussed this observation related to osmosis. Okay. Now, can you give me the inference for this observation? Pause the video for a while and try to think of an answer. Remember, you have to mention the final mass of the potato strip, sort of compared with the other results, as an example, and then explain at this concentration of the sucrose solution, what is the effect on the main RV, which is the final mass of the potato strip. Okay, and why is the final mass 2.5 grams? You can see that the initial mass is different from the final mass. Okay, pause the video for a while and try to answer this question. Let's look at the answer. Now, in sucrose solution B of low concentration, right, the final mass of the potato strip is higher than the initial mass, okay? 2.51 grams compared to 2.28 grams at the beginning. Because, okay, that this first sentence has no won't get you any marks, okay? Won't help you to obtain marks. But from there, because at a low concentration, the sucrose solution in B is hypotonic to the cell sap of the potato cells, okay? Meaning more dilute. And the water diffuses into the cells by osmosis, all right? Another mark. Thus, this increases the mass of the potato strip. Okay, so take note, you have mentioned the concentration of the sucrose solution, the low concentration, all right? So that's your MV, and you've related it to the increase in the mass of the potato strip, which is the main RV. Okay, now this experiment is found in activity 3.5, studying and determine the concentration of extracellular solution, which is isotonic to the cell sap of the plant, page 67. You can refer to that, and I also have one video on this. Next, let's talk about the hypothesis. The hypothesis links the main MV to the main RV from the objective. All right? So it's not the inference because you don't have to explain anything in hypothesis. Just how is the main MV and RV related? So in hypothesis, the three components are main MV, main RV, and the correct relationship. Now, what do we mean by the main MV and RV? These are taken from the objective or aim of the experiment that was stated at the beginning of the paragraph. Okay, sometimes the main RV and the sub RV is the same thing. All right, so it depends on the objective that is stated at the beginning of the experiment. Okay, so always refer back to the objective before you write your hypothesis, the objective that you underlined at the beginning of the experiment. So let's look at some examples. Okay, something more about the hypothesis. Now we know that the hypothesis should have these three components, MV, RV, and correct relationship. So there are two formats that can be used. The first one is for factors that can increase or decrease, all right? Then we use this format as MV increases or decreases, RV increases or decreases, or similarly, the higher or lower the MV, the higher or lower the RV, okay? So in this case, the value of MV can increase and decrease, and the value of RV can increase and decrease, okay? But there are some types of experiments that involve a few items that can be compared, like fruits and food samples, that we cannot say it increases or decreases. Okay, so we use another format. This goes for the two types of experiments. One is the energy value of different food samples, and the second one is the concentration of vitamin C in various types of fruits. Okay, they will use the second format. Of all the MVs, a specific MV1 has the highest or lowest RV compared to another two MVs, like MV2 and MV3. Okay, now let's take the example of energy value of groundnut. So this hasn't come up before, and vitamin C has already been asked in the first year of this KSSM syllabus. All right. So of three food samples, groundnut has the highest energy value compared to dried fish and dry biscuit. Okay. Now, if there are three food samples, so where is your MV and main RV here? Yes, the MV is the food sample. Okay, and then the RV, which is the main RV, is the energy value. Now, how do we calculate the energy value? We use a formula to calculate the energy value from the sub-RV, okay, using the sub-RV, which is the final highest temperature of the distilled water after the food sample is burnt, okay, the temperature of the water, the highest temperature of the water after the food sample is burnt. So that's the sub-RV, and the main RV, which we calculate from the sub-RV, is the energy value of the food sample. So what about your relationship here? Groundnut has the highest energy value. Groundnut has the highest, okay? But if there are only two food samples, then you have to change the English a bit, all right? You cannot say groundnut has the highest energy value, just that of the two food samples, groundnut has the higher energy value because only two food samples are compared, okay? So compared to maybe dried fish or biscuit or bread or something, right? Now, this is the other format that we discussed earlier. As MV increases, decreases, RV increases or decreases. So as temperature increases, the reaction rate of amylase enzyme increases up to an optimum temperature. the reaction rate of an enzyme decreases after the optimum temperature. So this hypothesis is for the experiment whereby we study the effects of temperature on the rate of reaction or on the activity, on the reaction rate or the activity of the enzyme amylase. Okay, it's written here. So this is another type of experiment, okay, where we use this different format. So if you find it a bit confusing, what experiment I'm referring to, look at the side here and do refer to the experiments that are discussed, all right? Or if you find that you have no time to revise all, then make sure that you go through the important experiments, all right, that are mentioned here. Okay, some of these important experiments. There are other important experiments too, okay, but these are some of them. Now let's discuss the table of variables. Normally only one or two variables will be tested because they'll probably give you one variable, ask you for one variable, and the method to handle the variable. Okay. Here we're going to give all three variables, discuss all three variables, and the three methods to handle them. So state the manipulated variable first of all, and you can get this from the objective of the experiment or from the procedure. And next, what's the method to handle the variable? You can use the format, use different MV, such as, and then state all the examples of the MV used in the experiment. For example, use water baths at different temperatures, such as 0 degrees Celsius, 20 degrees Celsius, 37 degrees Celsius, 40 degrees Celsius. All right? That is for experiment where we study the effect of temperature on the activity of amylase. Okay. Now for the energy value that we are of food samples, energy value of food samples, you say use different food samples, such as, now let's say using two food samples, then you must mention those two food samples, okay, such as cashew nut and a biscuit, okay, for example. And another way to put it is to change the MV, okay, change the temperature of the water bath, or okay, change the temperature of water bath. But for food samples, I think it's easier to say use different food samples. Okay. Now, next is the responding variable. Try to choose the sub RV, which you measure in the experiment or which you observe, or another word is ORV, is the same thing, the operational RV or the experimental RV. Now, as far as possible, don't write the main RV, which may only be obtained through calculation, okay, involving the sub RV. So if you write the main RV and there's some calculation involved, you have to give the formula of how you calculate the main RV using the sub RV. Okay, so it's easier to write the sub RV, and then after that you can use this format, either measure or weigh or count or observe and record, okay, the sub RV. And if there's apparatus used, then you say by using that particular apparatus. Okay, for example, for energy value of food samples, measure and record the final highest temperature of the distilled water by using the apparatus is the thermometer. Okay, now the textbook puts only final temperature, but I think it's better to put the highest temperature also, because in the procedure they have this word highest temperature, but in the table they just put final temperature. Okay, so measure and record the final highest temperature of the distilled water by using a thermometer. So it depends on your question. If the question just say final temperature, then just follow it. Follow whatever the procedure tells you to do. Okay, use the same terms. But actually it's the highest temperature, okay, when you're burning the food sample. So here we have the word measure and record because we are going to measure the temperature with a thermometer. Okay, and your sub RV is the final highest temperature of the distilled water. Now another example is when we are determining the effect of concentration of sucrose solutions on the mass of the potato strip. So we are going to weigh the final mass of the potato strip. So we cannot say measure and record. Now it's weigh and record the final mass of the potato strip by using an electronic weighing scale. Right? Weigh and record. So if you are observing a color change, then it is observe and record. If there's a color change and no measurement is involved. Now, state the control of fixed variable. So the format used is to fix the control variable at a specific value plus units. For example, fix the concentration of the enzyme amylase solution at 1%. Okay, or fix the volume of the distilled water, okay, that's used in the energy value experiment. Okay, fix the volume of the distilled water at 20 ml. All right. So another way is to use this format, use the same. Okay, but normally we try to put it in this format. Fix something at a specific value. Okay, plus units. Fix the CV or the FV. So it's important to note that these two experiments are different. The groundnut energy value experiment, where we burn the groundnut or food sample and then record the final highest temperature of the distilled water by using a thermometer. Okay, measure and record the final highest temperature of the distilled water by using thermometer. As compared to the potato osmosis experiment, we don't burn the potato, we in fact just soak it in the sucrose solution of a specific concentration, of different concentrations, right? And then we weigh and record first the initial mass and then the final mass of the potato strip. So you don't burn the potato here, you just weigh and record the final mass of the potato strip after you have immersed it in the sucrose solutions, okay, of different concentrations. And you do this, you weigh it by using an electronic weighing scale. Okay, so for one experiment, there is the burning of the food sample. For the other, the food sample is just soaked in the sucrose solution. Okay. Next, we come to table. Draw or construct a table of the data that you have obtained. All right, so construct a table based on the results of data. The table has 3 or 4 components, depending on how many readings there are also. So normally it has MV, sub-RV, then the main RV is obtained through calculations involving the sub-RV, and then there are units also in the table. All right, write the units together with the titles for MV, sub-RV, and main RV. So plan the number of rows and columns in the table, okay, and you may be given the basic format of what you should put in your table, so you can follow that. Sometimes they give the guidelines. Now, write down the complete titles and units as stated in the question, right? For example, as I said, for the enzyme experiment, we don't just write time taken. We must write the full name, like full phrase, like time taken for the iodine solution to remain brownish yellow, okay? The full titles according to what is stated in the question. Do not shorten the titles. So title for MV or the MV column is usually on the left-hand side, okay? And then sub-RV is usually in the second or middle column. And then from there, we get the main RV, which is on the right hand, in the right-hand column. All right. So that may involve calculations or workings. If there is, please show the calculation or working. Okay, especially for the main RV. Now, readings and values should be all to the same number of decimal places, should be consistent. For example, if you have one reading that's 1.5, another 2.8, another 3.6, you cannot write 1.5 and then suddenly 2.81. That's to 2 decimal places already. So usually it's to 1 decimal place, usually. Okay, depends on the experiment. But if the value is very small, you may have to go to 2 decimal places and so forth. If the value or reading is very small. Now, units are written together with the table titles, not with the readings itself. Okay, now let's look at some examples. So here's an example of a table. Okay, the question is construct a table and record all the data collected from this experiment. 3 marks. Okay, so again, at the top here, we have all the titles like food sample, mass of food sample in grams. All right, this is you weigh the mass of the food sample by using the electronic weighing scale, right? And then quickly write down the mass. All right, and the number of decimal places must be consistent. For example, to 2 decimal places here for all the 3 food samples. Then initial temperature of water, okay, 30 degrees Celsius. Final temperature 73, increase in temperature, okay, just final temperature minus the initial temperature, and then energy value of food is in kilojoules per gram, okay, and all the temperature for the water is in degrees Celsius. So all these units must be written into the titles here, okay, together with the titles, not with the readings here, right? Okay, so here we have the MV, right? The MV is the type of food sample, and then we need to know the mass of food sample to calculate the energy value. And then here we have the initial temperature of water and final temperature of water. Okay, these are all data. Okay, some people pronounce it data, either way. So then the calculations are done, okay, here. So increase in the temperature of the water. That means final temperature of water, final highest temperature of water minus the initial temperature of water, we get the increase in the temperature of water. Okay, so that's one calculation, and the other calculation is to use the formula to calculate the energy value of food. Okay, so correct calculation plus consistently to the same number of decimal places, for example 2 decimal places for the energy value. Okay, so this is the correct calculation. It's 1 mark. Okay, correct data 1 mark. Titles and units, all this here is 1 mark. If you missed out even 1 unit, you will lose that mark. Okay, so make sure you write down all the titles plus units. Now in your Amali exam, the practical exam, you may not be tested on, you may not need to, find the energy value for so many food samples. Okay, so it may be only 2 food samples, for example. All right, or they may modify the question somewhat. All right, so here our main RV is the energy value of food, but the sub RV is the increase in the temperature of the water after the food sample is burnt. All right, so don't confuse that with other experiments. Increase in temperature of water is used here, okay, in the formula, which is actually mc theta per W. W is the mass of the food times 1000. That means divided by 1000. Okay, the whole thing divided by 1000 is to convert it from joules to kilojoules. All right, and 20 here is the weight of the mass of the water, the distilled water in the boiling tube. It's 20 ml, so it's equivalent to 20 grams. Okay, so remember, this 20 stands for the mass of the water, and the 0.58 here is the mass of the food, which I sometimes call the W. So mc theta, C is 4.2, mc theta per W, W is the mass of the food, and then per 1000, okay, the whole thing is divided by 1000 to convert to kilojoules. So try and remember the formula and how to use it. So this is the table for the osmosis experiment, where we investigate the effect of concentration of sucrose solution on the mass of the potato strip. Okay, so your MV is on the left hand side. As I said, it's the concentration of the sucrose solution. The unit is M. You must write it inside the title here. Now, initial mass of potato strip is in grams, final mass of potato strip is in grams. And then the percentage change in the mass is just percent. This is the unit. So all these units must be here. And so 1 mark is given for all these titles, okay, plus units. Here I also give the formula for the percentage change in mass, which is final mass minus initial mass times 100 over initial mass. Okay, and then another mark is given for the complete for correct data here, the MV and the sub RV, right? So here I've written data must be to the same number of decimal places, or the readings, like 2 decimal places here, and this one 2 decimal places, because you use the digital weighing scale, which is quite accurate. Okay, maybe to more decimal places, but you round it up to 2 decimal places here. And then the percentage change in mass using the calculation here, final mass minus initial mass times 100 divided by the initial mass. Okay, so you get the value of the percentage change in mass make sure that you show your calculation, right? It will help you to check and confirm your calculations too. So 1 mark for correct calculations, this type, this one, okay? Now this is to 2 decimal places, and this is a negative value, right? Means the final mass is lower than the initial mass. Here is another table to investigate the action or the digestion of lipids by lipase, all right? So there are 2 test tubes, one with lipase, one without lipase. This is the MV, the content of the test tube is the MV. And actually it's the presence or absence of lipase, okay? Then the RV is the change of the color of the phenolphthalein indicator after 5 minutes, all right? That's it. So it's quite simple. And here our results is that when lipase is added, the phenolphthalein indicator changes color from pink to colorless due to the production of fatty acids after the digestion of lipids. Fatty acids will cause the mixture to be acidic, and then the phenolphthalein indicator will turn color from pink to colorless. But without lipase, there's no reaction. The phenolphthalein indicator does not change color and it remains pink, right? So MV here, RV here. All right. Next, we come to the graph. So if you ask to draw a graph, so what are the points to note? First, write the titles on the y-axis, which is the vertical axis, and on the x-axis, which is the horizontal axis. So make sure that you write the right titles according to the question. And if there are units, please put in the units, as some students forget to put in units for the titles, right? Then the graph must have a uniform scale, all right? And then that means it's evenly spaced out according to the how long it is, according to the numbering on the scale. Now, evenly spaced out, then the points must be correctly plotted from your set of results from the table, and then join all the points freehand with a pencil. So this one need to be emphasized because for other subjects, they find the line of best fit, like for physics, I think. I don't know about chemistry. So biology has its own set of rules that are different from physics and chemistry. Okay, the rules are different. Just like, you know, difference between dishes like char kway teow, nasi lemak, and roti canai. They are different, right? So please go through the rules for biology, like what I've stated here, just before the exam, so that you don't confuse it with the other 2 subjects. The rules for the other 2 subjects, they are different. So normally for biology, we join all the points freehand. We don't miss out a single point after we have plotted it on the graph. Plot it and put an X on the point there. Okay, so you join the points freehand with a pencil to produce a continuous smooth curve. So use a steady hand to join the points. Okay, maybe there are 2 points or 3 points. Now, only use a ruler if it's a straight line. If you are very sure it must be a straight line, then use a ruler. Okay, but very rare. Normally, it's a continuous smooth curve. Do not extend the curve to 0 point. Some students like, to join the point from where they are, join it back down to 0. Okay, or join from 0 up to the point. No, cannot do that, because if 0 point, there's no reading for 0 point in your table of results. You cannot just join to 0, okay? Unless the reading of 0 point is in the results, right? Now, also do not extend the curve beyond the first and last points, all right? So you cannot extend or project beyond the points that are there. Just between 3 points, then that means the curve starts at point number 1, goes to point number 2 also, and then stops at point number 3. If there are 3 points. Now here's the bar chart. So what are the components of the bar chart? Firstly, the scale. Okay, let's look at this one. Now in this case, the bar chart is the bar chart showing the energy value in kilojoules per gram against the food sample. All right. So the scale must be large and uniform. Fill 3/4 of the graph paper. So don't draw small size bars here, the mini bars, because then the marks will also be mini or small or less. So titles. Now the MV is usually is always on the horizontal axis. For example, food sample is your manipulated variable, and the RV is on the vertical axis, for example, energy value. Okay, so units must be shown here. So if your titles and units are correct, then you get 1 point or 1 mark. All right. So take note now to write in the units, because some students forget to write the units. Next, units write together with titles, and then the points are plotted correctly from the table here, from the table of results. All right, for groundnut, dried fish, and dry biscuit. So write down the names for the food samples below here in the MV. All right, and then bars. The bars must be separated from each other, equally separated. Okay, maybe 1 to 2 cm from each other, and then must be of the same width, right? For example, 1 cm, let's say, right? And then if it's a curve, it must be a smooth curve, and all the points must be joined freehand, and do not extrapolate beyond the points. We'll discuss that later. And histogram, where the bars are stuck together, is for continuous variation, all right, which is a Form 5 topic. So 1 tick for the titles and units here, and then 1 tick for the 3 bars that are separated and of the same width, and 1 tick for correct heights of the bars. That means by plotting the bars using these figures here. Okay. So here is a graph. Let's read the question. Based on the table of results, draw a graph of the rate of enzymatic reaction against the pH of the buffer solution. Okay, so this is not a bar chart. Okay, so just say draw a graph. Now, take note here. Rate of enzymatic reaction against the pH. So, rate of something against something is always the rate of RV against MV. All right. So the RV is on the vertical axis. Think of the word A here, just next to the RV. A is for atas. Think of atas. So it's the vertical axis, and this is on top, and then below that is the MV on the horizontal axis. Okay. So here we write rate of enzymatic reaction, and the unit is not given in the question. So you have to write your own units for this one, which is 1 per minute. Okay, and then 1 over T, and then for your MV is the pH of the buffer solution. Please underline, you need to write the full name, not just pH. It must be pH of the buffer solution according to the question. Okay, so don't just base it on this question, but what question you get in the exam. You must follow the questions, okay, accurately to the dot, and copy down the full phrase. Okay, copy like copycat, pH of buffer solution, exactly. All right, because some students just put pH. That's wrong. Okay, so here is our table of results that you have calculated earlier. So you can see the rate of enzymatic reaction is 1 over T per minute, see the unit. So plot these 3 points here on the graph. So you have to be careful so that the scale is large. And to do that, you have to put the variable symbol here, the zigzag symbol, all right, so that you can start with 4 here. Not start with 1, 2, 3, 4. Or you start 1, 2, 3, 4, then your scale for 5, 7, 5 to 9 will be very small and narrow, all right? So you can avoid the first part now, 1 to 3, by putting this symbol here. Okay, and start with 4, 5, and 6, 7, 8, 9. Okay, then it'll be large. And then the rate of enzymatic reaction, all right, the values are here. So it's plotted from 0 till the highest value is 1.4. Okay, so make sure you spread out make sure that the scale is large enough, okay, to fill about 3/4 of the graph paper, and that is uniform. Titles for MV and RV, okay, here you can see rate of enzymatic reaction against the buffer solution, pH of buffer solution, and the unit is here, right? Then points plotted correctly, so put an X for the points, exact location of the points, and then curve. This is a curve, so use freehand. I mean, draw the curve freehand using your hand, steady hand, and join all the points. Okay, don't miss the points. All right, don't find the line of best fit here. Just join all the points to form a smooth curve, And do not extrapolate down the point. So you can see here that I did not join this point here to 0, and neither did I extend beyond this point, okay, beyond pH 9, okay, because I only did the experiment until pH 9, okay, so I cannot join it to 10, extrapolate beyond that, okay, that's wrong, all right. So if you extrapolate, you will lose 1 mark for that, all right. Now let's talk about another question. Based on the graph that you have drawn, explain the relationship between, it could be MV with the sub RV, or MV with the main RV, or sub RV with sub RV, and so forth, right? Relationship between two different factors or variables. So first we have to state the relationship between the two components that are mentioned, such as MV with sub RV, or with the main RV, and so forth. So this is R, the relationship, okay, 1 mark. So the relationship is similar to hypothesis in this sense, all right? But you have to go beyond that, and you have to give two explanations. So for the explanation part, you can divide the graph into the different parts and then explain it, okay, as we will see afterwards. So give at least two explanations to get 2 marks. Now the explanations are quite similar to the inference in some ways, all right, that you have answered earlier. And the explanations should be based on biological reasons or the theory of in biology, theory of biology. So let's look at an example involving enzymes. All right, let's see how to explain this graph, right? So this is a bar chart, okay? It's a bar chart. So read the question carefully. If you're asked to draw a bar chart, please don't go and draw a line graph. So based on the bar chart, state the relationship between the food samples and the energy values. Then explain your answer. So this question comes after the drawing of the graph, and there are 3 points. The first one is R1, the relationship is similar to the hypothesis, okay? So of the 3 food samples, groundnut has the highest energy value, followed by dried fish, and lastly dry biscuit. See, it's similar to the hypothesis, because it is, hypothesis is actually the relationship between the MV and the RV, okay? MV is food sample, RV are the energy values of the food samples. So the next point is you have to explain why this is so. Why does groundnut have the highest energy value? So we go back to the inference now. Okay, so this part, when you need to explain the relationship, in the when you look at the graph, you have to explain the relationship. This type of question is related to the hypothesis and the inference, which you have already formulated, or you have already answered earlier, all right, in the first part of the questions. So just combine them together and maybe add a little bit more if you need to, a few more points. So E1, now this is the inference actually, no? This is because groundnut contains the highest lipid or fat content of all, followed by dried fish and then biscuit, highest lipid content. And thus, when the groundnut is burned, it will release the most heat, okay, when it is burnt completely. All right, so 3 marks there. As you can see from the graph, the groundnut has the highest energy value, followed by dried fish and dry biscuit. So let's answer this question. Based on the graph, state the relationship between temperature and reaction rate of amylase, and then explain your answer, 3 marks. Let's say we have drawn a graph of the reaction rate of amylase, or the rate of reaction of amylase against temperature. All right. So now we are going to state the relationship. So you look at the graph, it's bell-shaped. Now your MV is temperature, and you're studying the effects of temperature on the rate of reaction or reaction rate of amylase, right? So you can see that the graph is bell-shaped. And here there are 5 points. So the at 5 different temperatures, the rate of reaction was determined. Of course, in our practical exam, the time is too short to do 5 MVs. Usually it's about 2 or 3 temperatures only, right? Okay, now, so our relationship is something like, the relationship is something like the hypothesis. Now, as the temperature increases, okay, from low temperature to 37 degrees Celsius, the reaction rate of amylase increases up to the optimum temperature of 37 degrees Celsius. And after that, the reaction rate decreases again. Okay, so as MV increases, as temperature increases, the RV or reaction rate of amylase increases up to the optimum temperature of 37 degrees Celsius. Next, we have to explain our graph. You have to explain your graph, explain your answer. So you can explain the different parts, the low parts, and then at the optimum temperature, what happens, and then after that. So at lower temperatures, the reaction rate of amylase is low because the enzyme amylase is inactive here, right? Not so active, right? But then at the optimum temperature of 37 degrees Celsius, the reaction rate of amylase is the highest because amylase becomes the most active and there are the most number of collisions with the substrate molecules, which is the starch, leading to more reactions occurring. Then at higher temperatures like 50 or 60 degrees Celsius, the reaction rate of amylase decreases again, okay? Because now the enzyme amylase is denatured by the heat. So the heat will break the chemical bonds, cause active sites in amylase to change shape. So they no longer bind to the starch molecules, right? So that is the answer. So at least two E's here, E1 or E2 or E3. Now E3 is a bit long, so if you just mention that the enzyme amylase is denatured and the active sites in the amylase molecule change shape, could be enough, okay? Now, operational definition. State the operational definition. Operational definition means the definition that's based on the experiment. So you must mention your sub-RV, all right, and your MV. Okay, so the 5 components of the operational definition are definition, the basic definition, which you can also get from your bio textbook, okay, from the concepts of biology. And if there's any organism, you have to name them. Okay, for example, if potato strip is involved, the potato is involved, you have to mention potato strip, okay, for osmosis experiment. Now for energy value, you have to mention the groundnut or the fish or the biscuit or something else, bread or so forth. And then the RV, now to state the RV, you can put after the definition, which is shown by the sub-RV. This is a sub-RV. And then affected by MV. So RV is affected by MV. Okay. So my memory tip here is DOREM. Like I said, like Doraemon. D for definition, O for organism, R for RV, but it is stated in this format, shown by. It is shown by the RV. Okay. And then affected by MV. The RV is affected by MV. That is you can make it as a separate statement also, right? Okay, let's look at some examples. It's quite easy once you know DOREM. Okay, so based on the results of this experiment, define operationally the energy value of food. Okay, this is from my video on energy value. So can you try and write down the operational definition? Now, to know the operational definition, you must have a table where you have defined, you have determined what is your sub-RV, right? And also the MV, okay, which you have underlined at the beginning of the experiment, right? First of all, what is the meaning of energy value of food? It's the amount of what? Energy released, right? When 1 gram of food is burnt, right? So the food, you must mention the 3 types of food. Okay, try and write the operational definition based on DOREM, and we will discuss the answer in a short while. So pause the video for a while. Okay, let's look at the answer. The energy value of the food. So you must start with the term that you're going to define. You cannot say suddenly right here, so the heat released from 1 gram of the food sample is called the energy value. Cannot. You must start from the word that you're going to define. The energy value of food, according to what is in the question. If you just put energy value, then you have to write energy value in it. Okay? So follow the question. Now, energy value of food is the heat released when 1 gram of groundnut, dried fish, and dry biscuit are burnt. So here we have mentioned the definition. Heat released when 1 gram of the food sample is burnt, but it's good to write down the 3 food samples. You can put when 1 gram of food sample, bracket groundnut, dried fish, and dry biscuit are burnt, right? And then to write in the RV, put and it is shown by the increase in temperature of 20 ml distilled water. That is your sub RV, right? Then the energy value of food, all right, as in the definition, is affected by the type of food samples, the type of food sample. Okay, so that is your MV. Now here I also included the CV, all right, which is optional in this case, but it's more complete. Okay, so that's it. Let's look at another operational definition. Right, let's look at another operational definition. Based on the experiment, state the operational definition for hydrolysis of lipid. Okay, so this topic is found in this video, the Johor SPM trial, paper 3 for 2023. Right, you can refer to this video, which is quite new, if you like. So try and state the operational definition for the hydrolysis of lipid. All right, where we discuss in this experiment, we discuss the digestion of lipids by lipase. All right, and our responding variable is to observe the change in the color of the phenolphthalein indicator, okay, after a certain period of time, like say 10 minutes or so. Okay, try and write down the operational definition yourself. You must know the MV also, right? So pause the video for a while. Okay, let's look at the answer. Firstly, start with the term that you are asked to define, hydrolysis of lipid. Okay, so don't start with, the breakdown of lipids occur during the experiment. It must be the term first. Hydrolysis of lipids is the process of breakdown or digestion of lipids into fatty acids and glycerol, and is shown by the change in the color of the phenolphthalein indicator. The hydrolysis of lipids is affected by the presence of the enzyme lipase. Now, can you try and mark this and see whether it has all the points? Which are the various components that we require for operational definition? Where are the components? Okay, let's start with P1. What's the important keyword here? The definition, right? The basic definition is that the hydrolysis of lipids is the process of breakdown of lipids. Okay, the definition into fatty acids and glycerol. Okay, we mentioned fatty acids because it's the one that's going to cause the color change in the phenolphthalein indicator from pink to colorless because of the acidic condition that the fatty acids create, right? So phenolphthalein indicator will turn from pink to colorless. So the second part is the RV, and it is shown by the change in the color of the phenolphthalein indicator. So change in the color of the phenolphthalein indicator is your sub RV, and we must mention the word shown by, right? Then lastly, the term that you're discussing is the hydrolysis of lipids. So right there, the hydrolysis of lipids is affected by the presence of lipase. What is that component? Presence of lipase is the MV. Okay, when the lipase is present, then only there's the color change of the phenolphthalein indicator from pink to colorless. Okay, so prediction. This is the next question. You're supposed to predict the outcome or the results of the experiment if something were to change, right? So if something were to change, then you're asked to predict, then you must state how the RV or result would be changed. For example, it may become higher or lower or more or less than the original result or outcome. Secondly, you can suggest a reading or value as an example, okay, a specific value. We'll look at some examples afterwards. Write a complete sentence, not ayat tergantung. For example, some people, for prediction, they write, straight away they write less or higher, more or less or something than a particular result. They don't start with the what would be, the RV would be higher or lower or what. They just say less than something. There are students that write like that. So that's called ayat tergantung. It's not complete, so you won't get the mark. Now, give 2 or 3 explanations to explain your prediction. Okay, so you have the prediction and then explanation number 1 and then explanation number 2, E1 and E2. Now, explanation may be related to how the change affects the main RV. Okay, let's look at some examples to understand how to give explanation. So let's look at this example of a prediction question for energy value. So the diagram shows the cross-section of a coconut. The experiment is repeated with a small piece of the kernel from this coconut, okay? And the kernel is dried kernel. So the kernel is the white part of the coconut, the white flesh, which we usually scrape and then squeeze the santan out from, okay? And that santan contains a lot of lipids, right? So here you're asked to predict the energy value of the dried kernel compared to the dried fish and explain your answer. Now, compared to dried fish, or maybe compared to biscuit, okay? So we know for sure that the dried kernel will have a higher lipid content than biscuit or dried fish, okay, which has less lipids, especially not the oily fish, okay, certain types of fish, okay, that are non-oily. So we can state that the energy value of the dried kernel, okay, because you're asked to predict the energy value of the dried kernel will be higher than 2.24 kilojoules per gram, which was earlier determined as the energy value of the dried fish. Okay, there was an experiment earlier where we already determined that the energy value of dried fish is 2.24 kilojoules per gram. Okay, and we can give example. For example, the energy value of the kernel could be 3.00 kilojoules per gram. Okay, but the most compulsory part, important part is the energy value is higher or lower. In this case, it's higher than the value of the energy value for dried fish. Okay, than the energy value for dried fish. Number 2 is the explanation. So first part, now we are going to connect or relate the energy value, all right, high energy value to the MV. Okay, so the MV here is the dried kernel. It has a higher lipid content compared to dried fish. Okay, and also biscuit, but here it's compared to dried fish. So this causes the kernel to release more energy when it is burnt. Okay, because of its higher lipid content, lipid releases a lot, the most energy when it is burnt or when it's oxidized in the body compared to carbohydrates and proteins. Okay, lipid releases the most energy. So 1 mark for each explanation here. Another question on prediction, but this time it's a HOTS or KBAT question. It's not directly related to the first experiment that we do, okay? But it is still related in the sense that it involves osmosis, okay? So a boy is bitten by a leech. His mother puts some salt on the part where the leech is attached to the boy's leg. Predict what happens to the leech. So this topic concerns the concept of osmosis, all right? Now, so prediction is that when the salt is put on the leech will die and drop off from the boy's leg. So what actually happens? The salt forms a solution which is hypertonic to the cytoplasmic fluid of the leech cells. Okay, 1 mark. Water diffuses out of the cells by osmosis. The leech loses water, becomes dehydrated, and its cells shrink and becomes cremated, so it will die. Okay. So this is talking about the effect of hypertonic solution on the cells of an organism. Cause water to be drawn out from the cells. Next, we are going to classify the items. And to do this, you will probably need to complete a table based on the criteria that's given for classification. Let's look at some examples. Classification. Now let's look at this question. This could be related to energy value. Okay, so the following food items are sources of lipids. All these are sources of lipids. Classify them into 2 categories according to the type of fats that they contain, which are either saturated fats or unsaturated fats. Okay, so the table is constructed for you, and you have to fill in the blanks here, okay, with the right type of fats. Okay, so try this question first. Pause the video and try the question. Okay, let's look at the answer. Unsaturated fats. Which ones are unsaturated fats? Unsaturated fats are the healthy fats that do not cause a rise in the cholesterol level. Okay, so they are usually the vegetable oils, and their sources are from can be from nuts or fruits like avocado, okay, and vegetables, yeah, vegetable oils. So examples will be avocado, olive oil, sunflower oil, and then we have salmon. Okay, salmon contains healthy fats, the omega-3 fatty acids, right? And it is an exception when you compare to other uh, sources of animal food, okay, animal meat. Fish, the fish called salmon, contains the good fats, the unsaturated fats, okay? The rest are the saturated fats, and these are chicken fat, yogurt, milk, and ghee. All this comes from the animals, Okay, the following items are used in the experiment to determine the energy value of food samples. Classify them into apparatus and materials. Okay, so this is an old question from the exams before that, and here we have to be able to classify the items as either materials or apparatus. Now let's talk about apparatus first. Apparatus are all those hard equipment like glassware, right? Metal, plasticware, and so forth. Okay, that can be used over and over again. All right. So they include the retort stand, measuring cylinder, a long needle, electronic weighing scale, Bunsen burner, the barrier, the boiling tube, and thermometer. Okay. Now materials, on the other hand, are usually the stuff that the items that can be consumed, can be used up. All right, and then you'll need to get new ones if you want to repeat the experiment later on. Okay, so they include the food substances like cashew nut, fish, and so forth, nuts, and biscuits, and so forth, and cotton wool also, plasticine, because all these after you use you throw away, right? So when you throw away, it means it's consumed, it's used up already. And so these are the materials. And then distilled water and reagents are all materials also. Okay, so it's good to be able to distinguish between apparatus and materials. Right, another question on classification from the topic on osmosis. The following types of fluid have different effects on humans. Classify them into hypotonic, hypertonic, and isotonic solutions for humans. That means in relation to humans. Okay, seawater, tap water, saline drip, spring water, 2% salt water, and baby's tears. Okay, so try this question yourself first. Just classify them into the three types of solutions, and we will discuss this in a short while. So pause the video. Right, let's look at the answer. Now first you must know what is saline drip. What is saline drip? Saline drip is an isotonic solution of salt or sodium chloride that is dissolved in distilled water and is used in hospitals. So some patients are put on drip, saline drip, and it's actually 0.9% saline solution, which is isotonic to our body fluids like blood and also tissue fluid. Okay, it's isotonic to that. So saline drip is usually put into a person, okay, through the drip. It's dripped into the person, a sick person, to help him rehydrate. If he's suffering from dehydration, he will then be able to, the condition can be reversed. Okay, so let's look at the answer. The tap water and spring water, they are both hypotonic solution, okay, because they contain less mineral salts compared to our body fluids like blood and tissue fluid. Now 2% salt water is already hypertonic because our the saline solution that's 0.9% is the isotonic solution. Okay, so anything greater than 0.9% saline solution is hypertonic. That means it is more concentrated than the body fluids. Okay, and seawater definitely has a lot of mineral salts in it, so it is hypertonic. Now the isotonic solution are baby's tears and the saline drip. Okay, so that's all for this lesson. Thanks for viewing. Please do share, like, and subscribe. And all the best for your practical exam. Stay calm and study. Goodbye for now.